It is great to see such a large group of doctors interested in medicinal cannabis. And I'm very happy to announce that the next 2 hours of this symposium have been accredited by RCGP, Category 2 QIN CPD points, 4 points. It has been a great pleasure to work with RCGP team, and I want to acknowledge them for their commitment, enthusiasm, and support, and being so open-minded. So the accredited part of this symposium begins now, and it will be concluded by Dr. Ted's presentation after 2 hours. What we are experiencing now is such a large interest worldwide in cannabis, and not only as a medicine, but also so many other amazing properties of this plant. I can appreciate that it might be difficult for us doctors to embrace it, because after all, it has been labeled as dangerous and criminal for such a long time. And the public has been captivated by so many stories of healing. So now what we will experience more and more pressure and push to embrace cannabis, learn about it, and prescribe. And I find myself quite fortunate that I actually practice in New South Wales, because the process of prescribing has become much easier since mid-March. Until March, it was very difficult to obtain medicinal cannabis for general practitioner. It was convoluted, difficult, and very frustrating. And the main obstacle in obtaining medicinal cannabis by general practitioner was the need of endorsement from specialists. And most of the time, the specialists were basically not interested. And I have a feeling that their views were basically just due to lack of knowledge and probably lack of understanding. So what happened since mid-March? The New South Wales government changed. It is now possible to obtain medicinal cannabis, and the letter of endorsement from specialists is helpful, but not necessarily essential. And I have obtained approval for 37 patients so far. My main focus is chronic debilitating pain, which is, as we know, due to many medical conditions. The results so far are very encouraging. I have patients who suffer from pain due to severe ulcerative colitis, multiple sclerosis, rheumatoid arthritis, Crohn's disease, chronic back pain, and so on. What I am observing now, within this short period of time and a relatively short group of, a small group of patients, that in general, patients are already reporting improved quality of life. They feel better, they feel more relaxed, they sleep better, and the pain perception has changed as well. A lot of them say, I still have that pain, but it doesn't bother me so much. My brain kind of registers this differently. Some of them also saying, I feel more motivated, I feel more focused. And I was quite surprised to hear from one of the patients where she said, my mind is not so foggy. I think better, and my thinking is more clear. Also, a few patients commented that their driving has improved, because they say, before I was on all of these drugs and I was in so much pain that I really felt like a zombie. Now I feel I am better, more focused, and more cautious driver. So far, I feel really quite rewarded to be a part of this process and witness the changes in people's life. Obviously, I'm not saying that cannabis is going to be panacea for everything, that it will cure everything, but it is certainly a very important therapeutic agent. And I can say confidently, based on my research and my even short experience so far, that we need to change the way we think about cannabis for just drug of addiction into an important therapeutic agent that should be used judiciously, responsibly, in a proper clinical setting, so we can minimize the side effects and optimize the therapeutic effects. I am quite concerned that still large number of patients in Australia have to resort to black market. It is estimated that perhaps at least 100,000 or more people go to black market. What it means, they are subjected to products of dubious quality, and also they're missing out on a proper medical assessment, management, and monitoring. So I'm going to start my talk about endocannabinoid system. It was named after cannabis plant, discovered in the early 90s, and a lot of people say that it was probably one of the most important discoveries in human biology. And very interestingly, I'm not sure about you, but I never knew about it. I never learned about an endocannabinoid system during my studies. I discovered that through my own research about two and a half years ago. So what is this endocannabinoid system? It is universal, omnipresent system of internal regulation, cellular communication, and homeostasis. The discovery of this system was a result of a very significant research since 1964. Oh, sorry, I missed that. I go back. So this system is present universally among animals, humans, and plants alike, and probably appeared about 500 million years ago and kept on evolving. And this discovery of the system was a result of a very significant research since 1964. That year, famous Israeli scientist, Professor Mechoulam, discovered delta-9 tetrahydrocannabinol, THC, the most famous, most abundant part of cannabis plant, responsible for its psychoactive effects. But psychoactive effects is just a part of the story. As I'm going to talk about it in a moment, it is also a very important therapeutic molecule. Discovery of THC led to further research, and over the years, hundreds of other cannabinoids were discovered, and among them, the most famous now, CBD, cannabidiol, the second most abundant, non-psychoactive, non-toxic, and highly therapeutic part of cannabis plant, non-addictive. And in the early 90s, scientists cloned our own cannabinoid receptors, CB1 and CB2. Our bodies, we know now, are densely populated by these receptors. In some very metaphorical way, we can say that we've been wired out for thousands of years to interact with the cannabis plant. And so we have CB1 and CB2 receptors. CB1, cannabinoid 1 receptors, are mostly present in the nervous system, particularly in the brain, spinal cord, peripheral nerves, but also some other peripheral tissues, such as adipose and connective tissue, liver, testes, uterus, and gastrointestinal tract. But what is very interesting, there's hardly ever CB1 receptors in the brainstem, especially in the cardiopulmonary centers. So this is probably why that there is no reported deaths due to lethal overdose of cannabis. Now, CB2 receptors are mostly present in the immune system, as well as muscle, bones, and liver. Now, after cloning of the cannabinoid receptors, CB1 and CB2, the question was asked, why do we have these receptors? Obviously, there must be some ligands that interact with these receptors. And a few years later, scientists discovered anandamide and 2-AG, our own endocannabinoids. So in some very simplistic way, which is not very accurate, but perhaps demonstrates the point, our bodies are producing our own marijuana. And it is actually quite fascinating to say that we are making the molecules that are similar in structure and the way they act with THC. So all of these discoveries, we've got cannabinoid receptors, endocannabinoids, led to the discovery of the endocannabinoid system. And the endocannabinoid system consists of three major parts: endocannabinoids, cannabinoid receptor, and enzymes. So endocannabinoids interact with CB1 and CB2 receptors, and we have enzymes which are responsible for synthesis of degradation of these endocannabinoids. The enzymes which are responsible for degradation are MAGL, monoacyl glycerol lipase, and FAAH, fatty acid amide hydrolase. So the system basically regulates everything, almost every physiological reactions in our body. It is responsible for neural development, neuroprotection, movement, psychomotor behavior, pain, cardiovascular immune function, metabolism, appetite regulation, stress, emotion, memory, cognition, sleep, digestion, inflammation, thermoregulation. So in short, to summarize, we can say that this system regulates eat, sleep, relax, forget, and forget function. Now, both endocannabinoids and THC interact with the cannabinoid receptors based on key and lock analogy, where the cannabinoids are the key and the receptors are the lock. I'm just going to briefly mention the receptor sites. This site, orthosteric site, is the active site of the receptor, the key and lock analogy site, where THC and endocannabinoids bind here. We also have the allosteric site, and the CBD binds to that site very, very weakly. So by doing that, it changes the conformation of the receptor and modulates its activity. And modulates the action of the ligands that bind here. This is one of the mechanisms how CBD modulates the psychoactive effects of THC, acting as negative allosteric modulator. So metaphorically, it can be compared to the action of the dimmer in the light switch, where the CBD acts like a dimmer, modulating the intensity of the light. In this respect, it modulates it down. Now I'm going to explain now the endocannabinoid system in action, how it works to maintain homeostasis. So the endocannabinoid system, how it works in the neuron. Here we have a picture of the presynaptic neuron, synaptic cleft, and postsynaptic neuron. Now, as we know, neurons communicate with each other through electrochemical impulses. So the action potential travels here to presynaptic neuron. Then, obviously, many things are happening, but among them, it causes opening of the calcium channel. Calcium influx leads to release of neurotransmitters, which are stored in the vesicles at the end of the presynaptic neuron. These neurotransmitters travel across the synaptic cleft and bind to receptors at the postsynaptic neuron. Now, if everything is in balance, the process is repeated and the communication travels across the neurons. But if the neurotransmitters are upregulated, if we have too much inhibition or too much excitation, that causes opening of the calcium channel on the postsynaptic neuron. Calcium influx leads to synthesis of endocannabinoids, anandamide and 2-AG, which are synthesized from the components of the cellular membrane. And these endocannabinoids, anandamide and 2-AG, travel back to presynaptic neuron. Then they activate the CB1 receptor. And activation of CB1 receptor causes closing of the calcium channel, and it stops the further release of neurotransmitters. So this way, the endocannabinoid system works like a thermostat, ensuring that there is not much either inhibition or excitation. Once this process is completed, endocannabinoids are transported to the degradation site. There is quite a lot of research going on how it happens, and it's not completely understood, but now recent research suggests that possibly there are some intracellular transporters, which are fatty acid binding proteins, which transport 2-AG to the degradation site in the presynaptic neuron, and 2-AG is metabolized by MAGL, monoacylglycerol lipase, and anandamide is transported back to the postsynaptic neuron and is metabolized by FAH at the postsynaptic site. So, to summarize, we can see that this involvement in homeostasis, but also the characteristic of endocannabinoids. They are neurotransmitters, but in contrast to the usual neurotransmitters, they travel back. That's why they are called retrograde messengers. They are also not stored. like other neurotransmitters. They are synthesized on demand and released as needed. So they play a very important role in bioregulation.